This is my presentation on dietary fiber, short-chain fatty acids, and insulin sensitivity, mechanisms and evidence from human studies. Insulin resistance is the primary driver of elevated fasting glucose, postprandial glucose spikes, cardiovascular disease, non-alcoholic fatty liver disease, and also other chronic metabolic conditions. Insulin resistance is estimated to affect approximately 9.2% of the global population. Recent trends in gut microbiome research are focusing on short-chain fatty acids as mediators of insulin resistance. So the purpose of my paper is to evaluate the mechanistic, experimental, and observational human evidence to examine how the short-chain fatty acid pathway improves insulin resistance, and to assess whether fiber-based dietary strategies may serve as therapeutic interventions. So short-chain fatty acids act as signaling molecules. They are produced by the bacterial fermentation of carbohydrates. They go through the pyruvate cycle, and primarily acetate, butyrate, and propionate are the ones that are involved in this digestive process. They help to reduce chronic inflammation and decrease visceral adiposity. They support beta cell function and insulin secretion. Low fiber, ultra processed diets reduce short chain fatty acid production and increase low grade inflammation. They disrupt gut metabolic signaling and promote insulin resistance. Whereas fiber-rich dietary pattern will increase short-chain fatty acid production and improve gut metabolic communication, enhance insulin sensitivity, and improve glucose uptake. You can see from the diagram the pathway of dietary fiber in the gut. It undergoes fermentation and produces the short-chain fatty acids, acetate, propionate, and butyrate. Some of the immediate effects are strengthening the intestinal lumen, especially the tight junctions, which is very important in the prevention of infiltration by viruses and bacteria and other pathogens, which can cause chronic inflammation in the body if it permeates the gut. We can see some of the advantages, systematic benefits of the brain, the immune system, and the metabolism on the right. Insulin resistance is influenced by the intestinal barrier. A loss of barrier function allows pathogen-associated molecule patterns called PAMPs to enter into circulation. You can see that happening in the diagram on the right, the normal gut with the normal healthy cells and the tight junctions holding them together, and then on the right is the result of inflammation and tight junction breakdown. It's allowing those PAMPs to enter circulation, which will trigger an immune response and the release of pro-inflammatory cytokines. This chronic inflammation disrupts signaling and contributes to insulin resistance, obesity, non-alcoholic fatty liver disease, and type 2 diabetes. So the health of the microbial barrier determines actually the gut's dominant microbial community. By providing a stable environment and a nutrient-rich substrate, that supports the growth of beneficial bacteria. The fermentation of non-digestible carbohydrates such as inulin, fructooligosaccharides, beta-glucans, resistant starches, increase short-chain fatty acid availability. And beneath the microbial layer, the epithelial layer regulates the permeability through its tight junctions. That's what you're looking at here. And the immunological barrier, including immune cells, antimicrobial peptides, and cytokines, protects against what you are seeing right here, which is translocation. So each layer of the gut is specifically designed to maintain the intestinal integrity. So this is a little bit more of a detailed look at the short-chain fatty acid pathway. You can see that it follows, it activates GPR41 and 43, which stimulates GLP1 and PYY, which increases satiety and improves glycemic control. GRP41 regulates energy storage and metabolism in the adipose tissue. It also influences the appetite suppressant PPY in the gut. It affects energy expenditure in the hypothalamus. GRP43 regulates fat metabolism, lipolysis, and inflammation in adipose tissue. It also responds to the short-chain fatty acids to influence GLP1 gut motility and barrier integrity in the gut. It regulates the inflammatory response of neutrophils and other immune cells and tissue. GLP1 is an incretin which increases insulin secretion. It also decreases secretion of glucagon from the pancreas. It slows gastric emptying and therefore improves satiety because we feel fuller longer. PPY is the satiety hormone, which slows digestion and helps to regulate cravings. So a 2019 randomized double-blind placebo-controlled crossover trial evaluated 3 fiber interventions in overweight and obese adults with elevated HOMA-IR levels. Participants were given 42-day intervals of each of the 3 phases. The first one was a inulin propionate ester fiber supplement, which delivers propionate to the colon. The second was inulin, and the third was cellulose. And each group had each 3 treatments. They were followed, each phase was followed by a washout period of several weeks before beginning the next phase. So the result of the trial showed that the IPE supplement, the inulin propionate ester supplement, was produced the best results in improving glucose sensitivity. And what you're seeing there on the graph is like a lower response of glucose. So the results of this study showed that the fiber type actually matters more than the amount of fiber, because you can see that inulin produced the least amount of results, or was the least effect. And then a 2021 Mediterranean-style diet intervention noted that a longer adherence led to greater improvements in insulin sensitivity and glucose homeostasis. In this trial, participants were split into a single meal group and then their their glucose was measured. After just a single meal, there was an increase in GLP-1 and PYY, which would be expected. There was a decrease in postprandial glucose and postprandial insulin secretion. But after 4 weeks, there was increase in fasting and postprandial short-chain fatty acids and short-chain fatty acid producing microbiota or taxa. Also a decrease in the postprandial glucose and postprandial inulin, but you can see that that the 4-week trial actually produced the most modulation in the short-chain fatty acids. In a 2024 cross-sectional study, this is an observational study, found that individuals with prediabetes and type 2 diabetes exhibited lower short-chain fatty acids, reduced short-chain fatty acid producing taxa, and higher HOMA-IR levels compared with metabolically healthy adults. HOMA is a homeostatic. It's just an assessment of insulin resistance. It stands for homeostatic model assessment of insulin resistance. So these findings suggest that the habitual intake of fermentable dietary fiber promotes short-chain fatty acid producing microbiota, enhances microbial diversity, and increases short-chain fatty acid availability and improves insulin sensitivity. In a 7-year 2025 Spanish cohort study of 5,007 non-diabetic adults adhering to a high-fiber Mediterranean diet, actually yielded some surprising results. Most short-chain fatty acid is measured in fecal samples, but here they measured it through blood samples, and they found that higher circulating baseline at the beginning of the 7 years butyrate predicted an increase in type 2 diabetes by year 7. So the people who had the higher levels of butyrate in their blood were the ones who had developed diabetes by year 7. So this is like the complete opposite of what you would see if you were measuring short-chain fatty acids that were expelled through the body. But apparently when systematic butyrate is evident, that may signal impaired short-chain fatty acid utilization or gut barrier dysfunction or hepatic overflow. So these could all be like early signs of metabolic dysfunction before the individual comes down with diabetes. So it may actually be able to serve as a biomarker of metabolic dysfunction and offer preemptive treatment just with a blood test to measure butyrate levels. The composition of intestinal microbiota is highly responsive to shifts in fiber availability, directly influencing short-chain fatty acid metabolic pathways. Fiber-rich diets increase fermentable substrates and strengthen short-chain fatty acid metabolic pathways, and improvements in short-chain fatty acid taxa diversity could be seen within a day. So that's highly responsive. Mediterranean dietary patterns increased Prevotella, Bifidobacterium, fecal butyrate, and fecal propionate, while Western diets of processed foods decrease lactobacilli and butyrate producers. Those are important substrates for short-chain fatty acid proliferation. On the diagram on your right, you can see the de-evolution of a healthy intestine as it turns into leaky gut. The high fiber dietary pattern supports metabolic health by lowering A1Cs and reducing fasting and postprandial glucose, lowering inflammation, improving lipid metabolism, reducing fat toxicity, and ectopic fat accumulation. It helps to strengthen the gut barrier. It increases microbial diversity, short-chain fatty acid producing taxa, and enhances glucose homeostasis and insulin sensitivity. A large body of mechanistic, experimental, and observational evidence indicates that dietary fiber does play a central role in improving and maintaining metabolic health by modulating the gut microbiome, enhancing short-chain fatty acid production, strengthening intestinal barrier integrity, and improving insulin sensitivity. Mechanistic studies show that short-chain fatty acids, particularly acetate, butyrate, and propionate, reduce inflammation, enhance GLP-1 signaling, and support glucose homeostasis. Experimental human trials demonstrate that increasing fermentable fiber, especially soluble viscous fiber, improves insulin sensitivity, reduces postprandial glucose response, and shifts the microbiota towards short-chain fatty acid producing taxa. Observational studies consistently show that individuals with prediabetes and type 2 diabetes show reduced short-chain fatty acids and diminished short-chain fatty acid producing taxa, whereas higher habitual intake of fermentable fiber is associated with greater microbial diversity, improved insulin sensitivity, and lower metabolic risk. While colonic short-chain fatty acid levels reflect better beneficial microbial diversity, improved insulin sensitivity, and lower metabolic risk for type 2 diabetes, high circulating plasma short-chain fatty acids indicate impaired short-chain fatty acid utilization and serve as early biomarkers of metabolic dysfunction. Together, these findings suggest that increasing dietary intake of whole grains, legumes, fruit, and vegetables may enhance insulin sensitivity while decreasing fasting and postprandial glucose. Optimizing dietary fiber from fruits, vegetables, and whole grain carbohydrates can promote microbiotic taxa that produce short-chain fatty acids and improve glucose homeostasis. While high-fiber dietary patterns, including a traditional Mediterranean diet, demonstrate consistent metabolic benefits and highlight the importance of fiber-centered nutrition in type 2 diabetes prevention and management. And these are my references.